Today, again, we talked about mitosis in our lab last time, but today we're going to add on a couple of more phases with that. That's interphase, that happens before mitosis, and then at the very end of mitosis, right around telophase, is cytokinesis. So, what's this phase? You might tell me what this phase is? Prophase, right? How can we tell it's prophase? Because all the chromosomes are just kind of jumbled together. The nuclear envelope has disintegrated there. What's this phase?
Metaphase.
Metaphase. What's happening in metaphase?
They're getting ready to split.
They're going in the middle. All the chromosomes are going in the middle, getting ready to split apart, aren't they? So that would mean that this one is anaphase, where they split apart. Good, into sister chromatids. They split apart, go to opposite ends of the cell. So this one must be telophase. Good, good. Where it's about to start to split off in cytokinesis. Yes. First one's prophase. Yeah, prophase, because all of the chromosomes and all that genetic material are kind of jumbled up together because the nuclear envelope's broken down. Okay? So by the end of the lesson, we're going to be able to define the cell cycle. We're going to understand interphase and what happens within interphase. Understand mitosis and what happens with mitosis. It's important to understand that within this lesson, that interphase and mitosis are completely separate things. Mitosis happens second, and then interphase is going to happen before mitosis. All right, so just kind of keep that on your mind. We diagrammed mitosis last time in our lab. All right. Most of you, we weren't able to look under the microscopes in this class, but I'll show you guys some microscope slides so that you'll be able to see what it looks like under a microscope today. And we're going to define a somatic cell. What's a somatic cell? Okay. Honesty, right? Okay. So there are two kinds of cells in our body. We've got somatic cells and we've got gametic cells. Does anybody know what a gametic cell is?
No.
Not bad. That's actually good. Those are our sex cells, right? We've got the sperm and the egg, which are gametic cells, gametes. So then that would mean that somatic cells are all of our other cells in our body. So like our skin cells, stomach cells, red blood cells, things like that. Those are what somatic cells are. So we're going to be able to understand what a somatic cell is. Next week, we're going to talk about gametic cells and meiosis and the division of gametic cells. Right? So then cell division, which is, of course, mitosis. We're going to understand what a chromosome is. Does anybody know what a chromosome is? What is it?
It's parts of your genetic information.
There you go. They house our genetic information. Awesome. Okay. So daughter cells. What is a daughter cell?
It's the product
that are
It's the
exactly the same.
Product of, y'all are both right. Good. Product of mitosis. We make two identical daughter cells. Good.
So it's basically
Twins. Good. PMAT, we already know what PMAT is. Cytokinesis, which I mentioned just a little bit ago, where they're finally going to split off. They're going to pinch off into two cells. And then we're going to understand what regulators are and how we regulate our cell growth, because we don't want excessive cell growth, because that might lead to what? Cancer. And we know that uncontrolled cell growth leads to cancer.
Like cancer was
So like-
your own cells.
Yes, yes. Cancer is an attack on your own cells. It starts to replicate. We're going to talk about it.
So there's no such thing as an actual cancer cell. It's just your own cells.
Right, they're your own cells. They became cancer cells, and so they're replicating as cancer cells. So they invade your healthy cell at first, and then they start replicating as the cancer cell. Right. So the cell cycle is interphase, mitosis, and then cytokinesis at the end of mitosis, where it actually splits off. The first phase is interphase. We've got G1, where the cell grows. The cell's going to grow because the DNA is going to replicate in that S phase, the synthesis phase. That is the most important phase in interphase. This is where we're synthesizing our genetic material. We're copying those chromosomes. The cell's going to get really big, and it's not going to be able to function properly, so it has to split off. So that G2 phase is going to be the preparation phase for division. All right? That's called interphase. And then number 4, we divide to form two daughter cells, which are going to be identical in genetic material. And that is called the M phase or mitosis phase. And then we're going to begin the cell cycle again. Why would we begin the cell cycle again?
So that way
make
we
more
have
cells.
cells every hour.
So you can have more cells, right? So for example,
So
yeah, so that our levels can become normal again. So for example, if we were to go and donate blood, we would be given a lot of red blood cells, wouldn't we? Right? So our body has to make more red blood cells with this process so that our red blood cell levels can get back to normal. So we can maintain homeostasis. Remember that word? Long time ago? Yeah.
So when you donate blood, is your DNA basically in that person's body, or does it like basically become their DNA like after a while?
So we're going to get into the anatomy of blood in probably next semester. But what it is you've got specific types of blood, and if your blood type matches the donor's blood type, then the donor can give you that blood. If it doesn't match, then it's not going to work, and that blood's not going to be compatible, right? So we'll talk about blood type and things like that later. But again, G1, S, and G2 are an interphase. Again, it's not included with mitosis. That's happening before mitosis, okay? So again, as the amoeba sisters were talking about in our video, the interphase takes up 90% of the cell cycle. And then we've got the last 10% where mitosis is happening and it's actually dividing, right?
Right.
All right. So cell division, before a cell becomes too large, it divides, forming two daughter cells. Each daughter cell will contain a full set of genetic information that is identical. So if I said earlier, meiosis is the division of sex cells. The product of meiosis is 4 genetically unique daughter cells. This is the product of 2 daughter cells that are identical. Why are our sex cells dividing to make 4 unique daughter cells instead of the same? So if each one of them has different DNA, right? We want to have different DNA so that we can look different. If we were to divide our sex cells like we were with like mitosis, we would all look the same. We would all look genetically identical, right? So that's why we have 2 totally different processes, mitosis and meiosis. Meiosis divides into 4 genetically unique daughter cells so that we won't look identical to our parents, right? And we've got genetic material from our dad, genetic material from our mom, going together and crossing over to make those genetically unique cells. Yes?
But sometimes you end up looking like just like your parents.
You look a lot like your parents, but you're not completely identical. So, your question might be, how do twins happen? Do you guys know?
No.
No?
No.
So, at the embryonic stage of development, you guys were one cell at one time. Right? You guys were one egg. And so, whenever the egg was fertilized, you guys were going to be one. And then, at the very end, you guys happen to just split apart. Alright? So, you guys are identical, but you're not all the way identical, is that right? You guys are very genetically different, mentality-wise, too.
Oh, yeah.
Right? Okay. So that's how twins happen. Which is pretty cool that we have twins in the classroom.
Some twins just like, what is it called when they don't look the same?
Fraternal, those are 2 different cells in the same womb at the same period of time.
Okay.
Yes.
So that's the same thing about the triplets?
Yes, same thing. They split into 3. Right. So genetic information is passed on through our chromosomes. This is an electron microscopy of the telophase, looks like, doesn't it? Happening. So our chromosomes are thread-like structures within the nucleus. They, our human chromosome structure have 46 chromosomes. Now, to the right here, that's how we map out our chromosomes. That's called a karyotype. Does anybody know the significance of the 23rd chromosome? So we see number 22 there, and then that X and Y is the 23rd one. What is that?
Is that when it changes over with the parents?
Mm-mm.
And that's all the splitting apart.
Do what?
They share the same. Is it like a bad thing?
No, it's not a bad thing.
Is it all the way together?
Mm-mm. Okay. So The X and Y there, that 23rd chromosome determines if you're gonna be a boy or a girl. That's the importance. So, do you think this is a boy or a girl?
Boy.
I'm gonna go with God.
Boys, why are you saying it's a boy?
How
How do you know?
Hangs longer.
Why is it a boy?
Like, the first one's bigger than the second one.
Okay.
Yeah.
I mean, that's a good guess. Anybody else know?
The top's red.
Not quite.
Other than pink. I just.
No, we're not pink and blue in our bodies. So the X there, the X and the Y, that Y chromosome is the male contributor. That's how we know that this is a guy, because it has an X and a Y. Ladies have X and X, right?
What is that?
So we would have an X and X, similar to this here. That's the structure of the chromosome. That Y there is the male's contribution to determine if we're going to be male or female. If that Y is not compatible, then it'll be an X. You know, just depending on how that happens. Do it.
Yes, is that how the doctor does?
Yeah, we look at a karyotype and we can tell. So to the left here is the structure of a chromosome. We've got, it looks like an X there. The whole thing is the chromosome. And then the individual left and right sides are chromatids. Those are called sister chromatids. If we remember correctly from our lab, that's when anaphase happens. The spindle fibers are going to connect at the centromere, right there, pull apart and form those sister chromatids, right? Remember that? You're just saying, yeah. So, yeah, the spindle fibers are going to connect at this centromere here and pull it apart during anaphase, and that's what's going to make our chromosomes different. Have two different cells. Yeah. Okay? So, in interphase, again, it's G1, S, and G2. The S phase is going to be the most important, because what happens then? What happens in the S phase?
DNA replicate.
DNA is replicating, right. DNA is replicating during the S phase. Get it committed to memory that the S phase is the more important one of interphase. Interphase is not a part of mitosis though, all right? That's happening when? Before mitosis, good. So the cell's gonna grow in the G1 phase, DNA is replicating in the S phase, and then in G2, all of the cells going to prepare for mitosis, which is our division. So there is a red blood cell, kind of like what we were talking about earlier, that's going through interphase. All of this blue here, stained blue, is the replicating of DNA. Right? Again, interphase is not part of mitosis. It happens before. So, think tank. What happens in interphase? Talk to your group. You got about 30 seconds. 30. Talking about it?
It prepares for bodies.
Okay. That was weak. Alright, let's see. You're very excited. Fish, tell me what happens.
The cells grow. The cells grows and DNA replicates.
And then what?
Gets ready for cell division.
What are those phases called? Tree frogs.
G1 S
G1, S, and G2. Awesome job. Does everybody understand that part?
Yeah.
Interphase, happening before mitosis.
Yeah.
Good. So we're going to move on into mitosis now that we've gotten our cell ready to divide. We're actually going to divide. We remember PMAP, prophase, metaphase, anaphase, and telophase. This lovely, probably confusing picture is the cycle. We got prophase here. This is prometaphase. We're not going to go over that in this particular class because I would count it as metaphase happening. So then we move to metaphase where the chromosomes are lining up in the center. And then we've got anaphase where they're pulling apart, telophase where they're getting ready to cleave off and pinch off. And then we finally got cytokinesis where they turn into two cells. So in prophase, the chromatins are going to condense, which is getting smaller, right? And the centrioles separate and the spindle fibers begin to form. The spindle fibers, again, are going to attach themselves to the centromere, right? And they're going to get ready to pull them apart.
So do plant cells not have cell division since?
They do cell division, yes. And they have a different kind. So it's not exactly like an animal cell, they have a different kind of something that pulls them apart. And so the centrioles separate, but this is within an animal cell, okay? Cool. So this is what's happening in us right now, currently. The nuclear envelope is going to break down. Now to the right, you'll see a microscope view of prophase there. I know that you guys didn't get to look at the microscopes last time, but we did see a diagram similar to this last time. So that's what it looks like under a microscope. In metaphase, the chromosomes are going to line up across the middle of each cell, and each chromosome is connected to a spindle fiber at its centromere. This is a really good picture under a microscope of metaphase. You see it's lining up in the center there. Does everybody see that?
What is a centromere?
The centromere is going to be, remember the chromosome picture? It looks like an X. It's going to connect those two sister chromatids together. All right? It connects, it's like a little ball in the center of the chromosome. All right? The sister chromatids will separate into individual chromosomes and they're moved apart in anaphase. So you see here, they're being pulled apart to opposite ends of the cell, and then you'll see a picture under the microscope of anaphase. It's really distinct, you can see it happening. Pretty self-explanatory. You understand that part, anaphase?
I'll write it down.
Write it down.
Okay, you're good. And that plant cell on the right, is that how the plant cell looks?
This is an animal cell here.
Oh,
Yeah. It looks like a plant cell under here, so it's going to be an animal cell because it's splitting off. It's about to go back into the circular form. Yeah. Cool.
Cool.
Any other questions about anaphase? In telophase, the chromosomes are going to gather at the opposite ends of the cell, and they're going to lose their shape. And two nuclear envelopes are going to form around the sister chromatids that have separated. It's important to understand that the nuclear envelope reforms around, whereas prophase disintegrated the nuclear envelope, remember? And here we'll see a microscope view of telophase happening. And then we'll remember this diagram from the lab last class. Okay. And then cytokinesis, which is one that we didn't quite go over last time. The cytoplasm is going to pinch in half, and then each daughter cell, these are two genetically identical daughter cells that are forming. It's actually called the cleavage furrow, and we can tell why it's called the cleavage furrow. So it's going to pinch off into two genetically identical daughter cells. All right?
So is this after telophase?
This is, in a lot of times your professors will teach it in biology, like in college, as the same time as telophase. It's happening just at the very end of telophase. So this is, I'm teaching it as its own separate thing that happens, all right? Because it's important to know that it's not telophase, all right? Cool. So think of an acronym for PMAT so you can remember the phases. My acronym is Purple Monkeys Attack Tarantulas. And I want you guys to be able to tell me what happens during each phase. So talk about it with your group, think about a good acronym so that you can tell us what it is.
Pink
You can't steal mine.
Please. And then all together.
Tacos.
Yeah.
Pink monsters, eight tacos.
That's basically some purposes.
Pink mouse.
Telephones and
Alright. He volunteered to go first. What is your acronym?
Pink monsters, 8 tacos.
Pink monster. I've been having tacos all day. Can y'all think of anything other than a taco?
Toothpaste.
Toothpaste.
Whoa.
I like toothpaste.
Okay.
So what's happening in Proface?
Perfect. What is that taco?
Tell me what happens in Proface.
Proface separate.
Yeah. The spindle fibers form, and what's happening in that nuclear envelope? Disintegrates, breaks down. Good, awesome. All right, tree frogs, you wanna go next? What's your acronym?
Please, make
Please make a taco. I'll bring you guys some tacos next class. That'll be your reward. Please make a taco. What is happening in metaphase?
They line up in the middle.
Well, those chromosomes are lining up in the middle. Are they connected by a spindle fiber?
No.
They're not. They're connected at the centromere, the center of the chromosome. Good. All right. Do you guys want volunteers?
Rich, those working.
Rich, you got something?
Well, not really.
Not really? You guys got something? Go ahead.
People making animal tranquilizers.
Hey, dang.
Wow.
People making animal tranquilizers?
Shake up.
So last class I had one that said pink Mongolians ambush turtles. And I was like, okay, cool. So that's a good one. What's happening in anaphase?
Phase is whenever the daughter chromatids are gonna go off and start to separate.
Okay. Yeah, so they're splitting apart at the centromere, right? The spindle fibers are pulling those chromosomes apart and they form sister chromatids. Good. Do you guys know what's happening in telophase?
Telophase. The chromosomes gather opposite ends of the cells.
Are the nuclear envelopes forming again?
Yes.
Yeah. All right, they're getting prepared to separate, aren't they? Awesome. Did you guys think of one? All right, tell me one later, okay? So, awesome. You guys can tell me what happens with each phase. You'll be able to remember that more on the test now. So, cell cycle regulators. Regulators, the cell cycle is regulated by cyclins, which are regulatory proteins. There are two types of regulatory proteins. They're internal. What is internal? Inside, right? What is external? Outside. Good. Okay. So we've got internal regulators and external regulators.
Internals inside and externals out.
Mm-hmm.
Okay.
Yeah, pretty self-explanatory there. So internal regulators are proteins that respond to the events on the inside of the cell. These regulators keep the cell cycle in the correct order. For example, mitosis is not going to start until the DNA is replicated in the S phase. We're not just going to say, okay, we're ready to go now because mitosis is ready. Oh, but we haven't replicated yet. That's not going to happen because of these regulators. Okay? So again, mitosis is not going to start. Cytokinesis isn't going to go through until telophase is done. And you can just keep going back there. All right? An external regulator is a protein that responds to the events on the outside of the cell, right? For example, growth factors, they're the most important on the outside of the cell. They stimulate the cell growth and division of the cells, and they prevent excessive growth. If we didn't have these growth factors, excessive growth could happen. What does excessive growth lead to? Cancer. It could lead to cancer, right? So would you say that these internal and external regulators are important? Yeah. They tell us the order in which to divide and go, and they tell us when to stop
So
growing.
this happens after cytokinesis.
This is happening throughout. So remember those checkpoints that the amoeba sisters were talking about? This is the same thing. They have to go through certain checkpoints, make sure they have all their stuff together before they can move on to the next step. Okay? Yes.
I missed the last one. What was the last point?
In
These regulators keep the cell
Inside.
inside.
in the white order.
Correct.
Correct order.
In the video, it said that if it doesn't pass checkpoint, it will explode or something.
Yeah, the cell is going to die.
Wouldn't that hurt the other cells around it though?
No, it's not going to hurt the other cells around it because we've got specialized cells to come in and clean up all that cell debris. So if a cell is unhealthy, the macrophages is what they're called. They're going to come in and clean up that cell debris. All right, we're going to talk about that a little bit more later. We're talking about the division though right now. But good point. So, tell me what the cell cycle regulators do. About 30 seconds, give me a good definition of each one.
They make sure or they make sure it's healthy. What
We came up with one.
Did you?
Yes, I'll say it out loud.
Well, just a moment. I don't know.
So,
It says so.
Okay. Oh, gosh. All right. You have a PMAT? You
No,
not
we
ready
got it, we
yet?
got it.
Tell me what your PMAT acronym is from earlier.
Puppy making a tinkle.
Puppies
We
make
came up with it.
Puppies making a tinkle. All right. You're never going to forget it now.
That's a good one.
That was great. Excellent.
Took me forever to think of that.
I'm so proud of you. All right. Can you guys tell me what an internal regulator is?
An internal regulator is
Mhm.
inside the cell and it keeps it in the correct order.
Right. Good. Awesome. Trick rocks. Tell me what external.
I thought that.
It prevents it from
I'll put 'em on.
access growth.
Excessive cell growth. Good. It regulates outside the cell, doesn't it? Good. All right. Awesome job. So we've got one other point, which is uncontrolled cell growth. Uncontrolled cell growth leads to cancer, a disorder in which some of the body's own cells lose the ability to control their growth. Cancer cells do not respond to the signals that regulate the growth of most cells. So they don't respond to those growth factors that the external regulators are in charge of. Here you go. Okay? They divide uncontrollably and form masses called tumors, and then untreated cancer cells can break loose from those tumors and spread throughout the body. That's the difference between a malignant and a benign tumor. If it's malignant, they're going to break off and spread to other parts of the body. Benign tumors are going to just stay intact. So it's still a tumor, still cancerous, but it's not moving anywhere.
Doesn't it, if you cut a, like if a doctor cuts a tumor and tries to cut it off and this is part of it, wouldn't it spread to the rest of the body?
Yeah, it'll grow back, right? You're cutting those cancer cells loose, and so it'll travel.
that is
Anybody have any questions about cancer that they've been wanting to?
I don't know.
why is it so dead?
Because we haven't, of course, found a cure yet because we can't kill off each cancer cell. Because each cancer cell is individual. They're all different. Yes. Yeah, a lot of people die of lung cancer because it's really hard to target because you're targeting your lungs. Whenever you target, like on the video, whenever you're targeting a cancer cell, you're also killing the healthy cells with it. So that's why a lot of people who have cancer and undergoing chemotherapy and radiation are really thin and frail because their body's not making as many cells. They can't eat as much, things like that.
I heard somewhere on the news that scientists are like replicating white blood cells, like they're doing something to them to make them better and fight off cancer.
Yeah, so they're doing a lot of different things. They're introducing viruses to try to kill the cancer cells. They're trying to target those growth factors on the cancer cells that we were talking about. They're trying a lot of different methods to try to find a cure for it. So, yeah, hopefully one day we can find something to cure at least a few different cancers. So here, that's a tumor. Isn't that gross? That's what the microscopic view of cancer looks
Well,
like.
it's all like the seed.
Mhm.
The bottom one is something
one?
Yeah. Looks terrible. So here's a normal cell. We see that it's in a really good round shape. That's a normal animal cell.
Eggs.
The nucleus looks healthy, cytoplasm's full and intact there. Then we move over to this cancer cell here. And you see that this has 2 nuclei in it. And they're all irregularly shaped. They're gross. Nobody wants that compared to that. That's what those cancer cells look like in your body if you have cancer.
So is that what doctors see through
This is just a diagram of it, but you can see it on that previous slide. That's what the cancer cells look like under a microscope. A bat. So, Kate, tell me what cancer is. Just brief definition. All together.
It's an overgrowth of cells.
Overgrowth of cells.
Uncontrollable cells.
Uncontrolled cell growth. Good. Awesome.
Excessive cell growth.
Yeah, it's mean. They're fast. They're light and controlled.
Vicious.
They don't follow the rules.
Yeah, they don't follow the rules, right? Awesome. Okay. So, did we reach our lesson goals today?
Yes.
Define the cell cycle.
Yes.
Can we tell what happens during interphase?
Yes.
Is interphase mitosis?
No.
No, it happens when?
Before.
Before mitosis.
And
90% of the cell cycle is going through interphase. Awesome job. And we understand that mitosis, PMAT, and cytokinesis. And we diagrammed mitosis last time in our lab. And we now know the term somatic cell, cell division, chromosomes. What's a chromosome?
It's
Houses
the genetic information.
Houses are genetic information.
Yeah.
Awesome. And PMAT, daughter cells, that's the product, right? How many daughter cells do we produce?
26. Oh.
Two daughter cells are produced after mitosis.
Oh, I thought it was about chromosome. 4.
2, that's in meiosis. That's next class that we're going to learn now. But good, yeah. So cytokinesis, when it actually splits off, and we know what a regulator is. We know what that uncontrolled cell growth leads to cancer. Can we take a quiz over this next time?
Yeah.
Okay, and if you don't
Shut
understand
up,
something,
no.
if you can understand something, write it on your exit slip, all right? Go ahead and write it on your exit slip. I have a cartoon here that says, all right, now I get to have 2 girlfriends. Why does he have 2?
2 daughter cells.
Because it's 2 daughter cells.
Oh my.
Awesome.
That's funny. Hey,
Okay, so write on your exit slip, and we're about to play a game. You'll put all your notes away.
I'm good. One day. You're done.
All right, we're gonna play a game. It's called I Am. I'm gonna keep score. It may or may not be for bonus points. You just need to act like it is. All right. You guys come over here so that I can see you better. Yeah, y'all two come over here. You guys are gonna work together as a group. I'm gonna ask you guys a question, and it'll be presented up here. You have to knock and pound on your desk, and I'll acknowledge you. And when I acknowledge you, that's when you can give me the answer. If you speak out of that, if you answer without me acknowledging you, you're out of the round. Okay? It's going to be over mitosis, cell cycle, interphase, and cellular respiration and photosynthesis that we learned last time. All right, so are all your notes away? We can't use notes.
Oh, neat.
Okay, so for example, I'm a cell feeder. Of course, we know it's an autotroph, but you're going to go like this. I'm going to call on you, and then you say the correct answer. You get that point, all right? Again, may or may not be for bonus, so play like it is, okay? Everybody ready? Number 1, I'm the last step of mitosis. I divide the cytoplasm and create 2 daughter cells. Nope, not telophase. Still play. Cytokinesis.
Oh my, I forgot about that one.
Good job. All right. So cytokinesis is the answer. Good. So I am the time that occurs from prophase to cytokinesis. What is my name? Nope. Nope.
Mitosis.
There you go. Mitosis. Good job. I'm the third phase of mitosis. There you go. Good. Alright, guys. It's getting pretty heated.
Yeah.
I am the phase involved in G1. Oh, nope. There you go. Good.
If he knocks, can like all of us? answer?
Yes, all of you can answer.
Like-
Y'all are a team. You're a
Okay.
group, okay?
Oh, yeah.
You can stop.